So this is going to be an explanation of what your lab was over the last couple of days. I don't know why I'm doing all these things with my hands now. I'm just suddenly very self-conscious. All right, so let's go over this really quickly. The people at Biorad sell a product, a plasmid called P-Glow. I didn't want to have to buy it for everybody in this class. I was hoping that we could make it here. And so the way we were going to make it is by putting it inside of a bacteria and growing it up and then sucking it back out of that bacteria. So that was basically the first part of our lab. Now, as long as one or two of you were successful in this process, we're going to be fine. Not everybody had to, and the reason we did this lab was really not to learn this process, but to get you guys comfortable using a lot of the molecular biology tools and learning things like you have to write stuff down after you do an experiment.
Oh my god.
Okay, so what we did is we took the plasmid, we put it in a little bit of water. We put that water in a tube containing bacterial food and bacteria. So this is the bacteria, this is the plasmid, this is the food. The food is called LB, just so you know. It's luria broth. It's made up of ground up yeast. It smells terrible. All right? We put this tube through a process called heat shock, which means I take the tube, I put it in an ice bath, and then I put it back into a warm solution. And for some reason, that makes the membranes of this bacteria take up the plasmid. So now you can see the plasmid is inside this bacteria. The cool thing is that the plasmid has one little programming piece called the ORI. And the ORI is a programming piece. So when the bacteria copies its own DNA, if a plasmid has the ORI, it will copy the plasmid as well. So we grew this up in a shaker, a very high-tech shaker. That definitely was not duct taped together.
Should I get a photo of that?
No.
All right.
That definitely was not duct taped together. And we grew that overnight for about 14 hours. After the end of that, we put it into a tube and we added a lot of base to that tube. The base blew the bacteria up. It ripped apart the cell membrane.
No.
So now we have a tube with Jerry in it, in pieces, and plasmid. So we took all of that junk and we put it into a Qiagen Miniprep column. Qiagen is a company. It's Qiagen, just so you know, because I might call this a Qiagen column in the future. The Qiagen column is pretty simple. Actually, it's not. It's this little filter that they have is awesome. It has made life so much easier for a lot of people. But it's basically a tube with a filter in it. That filter has little parts in it that are positively charged. Little pieces of resin in it that are positively charged. So we run all this stuff through. And the super negatively charged items, the plasmid, will actually go through. Now, you might ask yourself, why doesn't the DNA, or does the regular DNA also get stuck in there? And that's something we can talk about later. But right now, in this column, we have the cell junk, or Jerry's junk, going through.
Poor Jerry.
Nope. Jerry's pieces going through, and the plasmid stuck in the filter. So what we then did is we washed everything out, and the filter has the plasmid attached to these little positive pieces. When you add water, the water, because it has these two charges, because it has electronegativity differences between the oxygen and the hydrogen, will interfere and get in between the positive charge and the plasmid. And if you keep adding water, eventually the plasmid will just fall out. When the plasmid fell out, we collected it in a tube, and that's what we nanodropped. So what we were really trying to see when we nanodropped was, did we get this plasmid out? How much of this junk did we get in there? Were there maybe some contaminants from the whole process? And that's why we were so concerned with nanograms per microliter, or the amount of plasmid, and then the 260-280 number around 1.8. The 260-280 number around 1.8 tells us that this is pure.
What is the 260-280 number?
So 260-280, so what does that mean? Basically, if you have a tube, or two tubes, and you shine light through them, some of that light will be absorbed. If you have DNA in that tube, more of that light will be absorbed. So you look at the light that's coming out, and what you'll find is that at 260 nanometers, basically a color, one color, and 280 nanometers, a similar color, you'll get absorbance. And that ratio will be around 1.8 if the sample has water and DNA in it. If it has other things in it, absorption changes. The absorption of those two colors changes.
It's an example of like something else.
You can go look there. There's a list on the wall there of all the different things. RNA, for example, will change the number to make it go up. So if we have RNA in there, the 260, 280 will drive up to about 2. But if we have a 260, 280 of around 1.8, we know we have a pure sample. And if we have a high nanograms per microliter, specifically anything above about 20 nanograms per microliter, we are successful in having isolated this plasmid from this original sample. That's what you did.